Well, thank you. Thank you for the invitation and thank you for being still there. So briefly, uh, these are the cells, you know them, which have been so far the most extensively tested clinically, if we start with the cells. And overall, I think we can say that safety has been fine, but contrasting with inconsistent efficacy so far. And as we are talking about safety, I just would like to draw your attention that we now have 54 clinical trials using pluripotent stem cell derived cells, including those from embryonic stem cells. And fortunately, so far, there has been no safety signal. So it's rather reassuring. So this is for the cells. Now, for the cell products, we now have 20 clinical trials which are currently registered, only one so far for the heart, patients with acute MI. The study should start early 2021 in the Mayo Clinic, and we could have a second study, which is actually our study, which hopefully should also start next year using cell products in patients with heart failure. Now, why are we talking about cells or cell products? Well, because of this progressive shift from remuscularization to paracrine activation of endogenous pathways. And this paracrine activation, we know, is largely mediated by extracellular vesicles, not exclusively vesicles, but largely vesicles, which once internalized into recipient cells can hit a variety of signaling pathways contributing to heart repair. And this has been shown for years since the pioneering works of Victor Zoe. Now, this shift from remuscularization to paracrine signaling is indeed strongly supported by the demonstration in a wide variety of preclinical models that the benefits of stem cells could actually be duplicated by the administration of their secretome. And I think it's more appropriate to use the term secretome enriched with extracellular vesicles than to restrict that to the exclusive exosomal fraction. Now, having said that, in a clinical perspective, and I think that was the objective of this talk, what could be the advantages of using cell products over cells? I'm not trying to say that cell products will work better, but if we assume equivalence, it might be, say, more user-friendly to use cell products, and there are a couple of arguments in favor of this assumption. Number one, dosing can be more accurate. Here you see that for cells to be effective, they probably need to be within a fairly tight range. And this is difficult to achieve with cells. All of us who have been working with cells know that there is a certain variability. And this is not the case with extracellular vesicles, because regardless of the metrics, you can just manipulate the conditions of cell culture until you get your target number. Number two, the large-scale production of cell products is closer to that of a drug. We know now, quantify vesicles. We have identity markers which allow to characterize them. We do have fairly robust potency assays which provide straightforward readouts. This is a survival assay. Here you stress the cells by serum deprivation and just can see the improvement in cell viability in the presence of the secretome in a dose-dependent fashion. And it's the same for the angiogenesis assay, where you see that in a poor media, if you add vesicles, you have a nice healing of the wound. And clearly the regulators are pretty happy with these kind of potency assays. And finally, with omics now, It's possible to map the cargo and predict the targets. Number three, which is important, the secretome can be cryo-frozen and is now available off the shelf. And in clinical practice, this is a real advantage. Here you see, and this is well known, that freeze-thaw cycles will increase cell mortality. This is not the case with vesicles. These are recent data from our lab. Again, you see here cells that have been stressed by serum deprivation. This is a positive control with FBS. And actually, you really rescue the cell and their viability with the EV, with the vesicles. But interestingly, you get the same kind of improvement if they have been freeze-dried, with or without the presence of threonose. Then you also can get a better distribution of the product. Why? Because when you inject cells, and unless you move the syringe constantly, cells tend to sediment, which means that there is a time-dependent variability in the amount of cells which are delivered per site. In other words, the first sites to be injected will receive more cells than the second, third, or fourth sites. And this is obviously not the case with a vesicle or a secretome suspension, which behaves as a Newtonian fluid. And finally, you avoid a couple of potential complications associated with cells, arrhythmias, and rejection. Rejection is important. Here you see the immune profile of the cardiovascular progenitors we derive from IPS. And you see that they do express a little bit of HLA-1 as well as some ligands for NK receptors. Now, if you look at the immune profile of the secretome, the EV-enriched secretome of the same batch of cells, they really look immunologically neutral, which is supported by the fact that they do not stimulate T lymphocyte proliferation, in contrast to the positive control. And likewise, they don't cause NK degranulation. So that said, they also raise concerns that need to be addressed. The first is the selection of the mother cells. And indeed, in the literature, you will find all cell types that have been used for producing the secretome, but there is compelling evidence that probably the best producers are cells which phenotypically are close to those of the tissue to be rescued, and number two are cells which are at an early stage of differentiation, because the earlier the cells, the higher the secretory profile. And for us, this is quantify IPS-derived cardiovascular progenitors as the ideal candidates. Number two, we do have to address the problem of the large-scale GMP-compliant production. Obviously, if you use cells, you go all the way through until you get your therapeutics. Now, with the secretome, we have to collect the conditioned media and obviously to purify it. In the clinics, the only method so far, which is probably the most suitable, is tangential flow filtration, because it is compatible with GMP facilities and because it allows you to handle very large volumes, which is important for clinical applications. This is the principle of a TFF with the membrane. And if you compare that with other methods that have been used for purification, you see that you lose a little bit in terms of recovery, but you can be very specific, and specificity obviously will depend on the size of the pores of the filtering membrane. And then you get your secretome and can freeze it, put it on the fridge, and use it on demand. And actually, we come to the third and challenging issue, which is the extent of purification. This is the TFF device that we use in our GMP facility. But the question is, to which extent should we purify? And it's not so evident that we should purify too much. First, because it's challenging. No one really agrees on the specific markers for isolating one fraction instead of the other. But more importantly, you may need actually all the ingredients of the soup to make it effective. And this is actually supported by a couple of studies like this one. It's a cardiomyopathy model, and you can appreciate that you get exactly the same kind of results with the cells or the whole condition medium, regardless of the endpoint you look at. I'd like to finish by addressing a couple issues which are common to cells and their product. The first has to do with regulation. We know that cells are ATMP, and ATMP has a very definite regulation. While interestingly, the secretome, the EV-enriched secretome, is not considered by the regulators as an ATMP. It's considered as a biological medication with a specific regulation. Having gone through both, I would not say that it's easier or that the regulation is looser with biological medications. But at the end of the day, my impression today is that it's a little bit easier to get through with biological medications compared with cells. Delivery, that should make Dr. Marvin happy. Why? Well, this is a study in patients with heart failure. It's an interesting study showing that if for one reason or another you stop medications, you will get recurrence of events, meaning that it's unlikely that with a single shot we can achieve sustained therapeutic benefits. So if we go for repeated treatments, as shown by Eduardo in his previous talk, if we go for repeated treatments, we need a non-invasive approach. That's the only means. And the only non-invasive approach is clearly the IV one. So the question is, if we give the cells or the products intravenously, is it going to be effective? Well, this is a summary of many preclinical studies using cells injected intravenously. Interestingly, most of them have reported improved outcomes. But these are experimental studies, and we know there are limitations. What is more interesting is that these encouraging data are now supported by the early results of the first clinical trials using IV delivered cells. Now, if we now look at the cell products, we don't yet have clinical data, but this is a meta-analysis of these vesicles, the secretome, in animal studies. What you see is that you get a benefit in terms of left ventricular function, you get a benefit in terms of reduction of infarct size. It's because these positive outcomes are achieved regardless of the route, whether it's direct intramyocardium or IV. And actually, in the 20 trials that I showed you previously, 6 of them are currently using the IV approach. Now, then the question is, how can it work? Because we know that the cells or their products are going to be sequestered primarily in lungs or spleen and liver. Well, to echo what has been said previously, it seems that a primary mechanism of action would be a systemic modulation of inflammation. And if you look at these studies with cells, you see that a lot of endpoints indicative of inflammation are shifted in the right direction, in the
Time's
direction of
up.
repair. M2 macrophages, increase of IL-10, and so on. Interestingly, this is the kind of results you get with IV cells, but you get exactly the same with cell products. In other words, it might be that a primary mechanism of action of the cells or their products is this systemic modulation of inflammation with remote effects on the heart, mediated by endogenous immune inflammatory cells whose phenotype has been changed by the cells or their product and which then travel through the bloodstream. Finally, a last word regarding the models, because cell therapy is really something specific. So far, we've always used the frequentist approach. That is, you do a RCT, and at the end of the day, you unblind the data and see whether it works or not. I think we should take some lessons from our colleagues in oncology using the Bayesian models based on predictive probabilities, and which allow you to integrate data as they accumulate, as the trial is moving, which is probably time saving because it allows you to eventually reframe the trial in one way or another to make it successful or to conclude that it will not. That said, thanks once again for your patience, and I'm ready to answer any question you might still have. Thank you.